Good afternoon or evening, everybody. It's evening for me, I guess. Hey, I'm here to review Unit 3 with you. So we're talking about enzymes, cell respiration, and photosynthesis. I've got the learning targets pulled up here, and I usually just walk you through the learning targets. But this time I decided that these learning targets are completely embedded within the notes that we've been going over, the prezzies that we've been doing. So I thought I would just review those prezzies today. I'll try to just get to the highlights and go as fast as I can. You definitely want to speed this up whenever I'm rambling about something that is not important to you. So the first part of the notes we needed to talk about, or that we did talk about, was the first and second laws of thermodynamics. So the first law, and I'm just going to skip ahead as fast as I can so we can see these. The first law just says that matter and energy cannot be created or destroyed, but that it can be transformed. And during this section, we talked about exergonic and endergonic reactions. You would need to be able to look at a reaction and be able to tell if it's exergonic or endergonic. Notice that with an exergonic reaction, energy is released, and the amount of energy stored in the products is less than the amount of energy stored in the reactants. An endergonic reaction is the opposite. An endergonic reaction shown here is where you have low energy reactants and you store energy. Energy is taken in to make products that have higher energy. The perfect example of this is photosynthesis. We start with low energy carbon dioxide, we take in the sun's energy, and we build these high energy glucose molecules. That's an example of an endergonic reaction where we've taken in energy. All right, I'm trying to think if there's anything else. I don't think there's much else on this page that we have to go over. So let's go ahead and take a look at the second law of thermodynamics, which is the one about entropy. Entropy is one of those words that it's so easy to get turned around in your head. So entropy means disorder. So it says any closed system will tend toward a state of maximum disorder, maximum entropy. For life, maximum entropy is death. Now that's a funny way of saying it, but for any living organism, when we hit our maximum entropy, we are dead. So it's true for the universe as a whole, but we are an open system that is constantly adding energy in to stop disorder, to stop entropy. And so we're taking in the sun's energy, we're taking in food energy, and that's how we are maintaining our organization and preventing entropy. Yeah, I feel like that's enough information, and we should be done with this one. I'm going to move on to enzymes. You know what? Why don't I double check? Let's look at the learning targets and see if there's anything I didn't mention. So they require an input of energy. Life requires a highly ordered system and does not violate the second law of thermodynamics. Cell processes may be coupled with cellular processes that require energy. That happens during both photosynthesis and cell respiration. You don't need to know anything about Gibbs free energy. That is something that comes up in chemistry, but we don't have to know for this. And we'll talk about this in the next set of notes, but it says energy-related pathways in biological systems are sequential to allow for more controlled and efficient transfer of energy. That's that whole analogy of your gas tank and dropping a match into your gas tank and letting it explode and not controlling the energy, or getting your gas tank and using spark plugs to just start a tiny explosion and control it so that we can move your car and hundreds of miles and not waste a ton of energy. So that's what they're talking about here. So we'll get to that more in a little bit, but just wanted to make sure that I covered everything in that. All right, I'm moving on to the notes on enzymes. So you probably remember the basics about enzymes. Enzymes are proteins, and they have very specific shapes, as all proteins do, and anything that changes their shape affects how well they work. This is a diagram right here that you absolutely need to become comfortable with. In the black line, you're seeing the amount of energy. It's called the activation energy, the amount of energy that's required to get a reaction to go from its reactants to its products. So under a normal circumstance, you put in X amount of energy to get that reaction to occur. But in the presence of a catalyst or an enzyme, you can put in less energy and the reaction will occur. And that's the benefit. And it allows reactions to happen sometimes millions of times faster than they would under normal circumstances. The really easy example that we had of that was the enzyme that was in the liver. It's called catalase. It reacts with hydrogen peroxide and it makes hydrogen peroxide break into water and oxygen. You can make hydrogen peroxide break into water and oxygen by adding energy yourself. You could heat the hydrogen peroxide, you could shake the hydrogen peroxide, you could add ultraviolet radiation to the hydrogen peroxide. All of those would make it occur. But by adding the enzyme catalase, it happens incredibly fast with very little energy. And that lowers the amount of heat that builds up in a body. So enzymes are able to do these super efficiently with very little heat being given off. All right, so let's talk about how enzymes work. Enzymes, again, have special shapes. They can bend a little bit around, and you need to know that the things that go into the enzymes are called substrates. So they can bend, it's called the induced fit model, and they can bend just a little bit. The active site is where this substrate moves in, and then most enzymes either put two molecules together or they pull a single molecule apart. In the example here, they rearranged some bonds within molecules. So they do lots of different things. All right, that's the induced fit model. Rubisco, we're going to talk about a little bit later. I'm going to scooch on because I don't think we need to know these things. Whoops, there was one we needed to look at here. Nope, we didn't. I lied. All right, let's talk about competitive and non-competitive. You definitely need to know this. So I think the diagram's super self-explanatory. Here's the normal substrate and the way it fits in, kind of like a bendy puzzle piece. If we're going to have competitive inhibition, so if we want this reaction to slow down, we don't want it to happen super fast, we can have another molecule that gets into the active site and gets in the way of the substrate, and that will slow down that enzyme reaction. So that's called competitive because they're both competing for the same spot on the enzyme. Non-competitive inhibition also slows down the rate of the reaction, but in this case, remember, try to pull all this stuff together. This is from biochemistry unit. Remember that proteins have all kinds of levels of structure. They have the primary, secondary, tertiary, and quaternary structure. So just a quick reminder, primary structure is all the amino acids put together. Secondary structure is the alpha helices and the beta pleated sheets and the hydrogen bonds that occur between them. Tertiary structure is the R group interactions. Remember, R groups are the part of each amino acid that's a little bit different, and some of them are polar and they're attracted to each other, and some of them are non-polar and they're not attracted to each other, whatever. And so here's my point, that a non-competitive inhibitor is another molecule that sidles up against the protein. And because of all those R groups react to this new molecule. And as they change their bonds and react, they change the shape of the active site. And now the substrate doesn't fit in it. So they're not competing for the same location. The non-competitive inhibitor slows the reaction by actually causing the enzyme to change shape and not fit with our substrate anymore. All right, moving on. Let's talk about allosteric sites. So an allosteric site is a site other than the active site. Now, that's what we just saw in the non-competitive inhibitor. Let me back up again. This would be considered an allosteric site. This is the active site. This is an allosteric site. Oops, I'm going the wrong way. There we go. All right, so in activation, it's possible to have a molecule that touches and it causes all of the parts of the enzyme, all the active sites to be ready. So they've activated. It says binding of an activator molecule can stabilize the enzyme in an active conformation so that you can basically ramp up the reaction and make it happen even faster. The opposite can happen. You can actually have inhibition where a molecule attaches in an allosteric site. Both of these are allosteric because they're not in the active site. In this case, the inhibitor changes the shape of the active site, and so now it slows down the reaction. So the cell is able to speed up or slow down the reaction using these allosteric sites and either activators or inhibitors. All right, and trying to see if there's anything else we need to know. Right, so the binding of a substrate molecule on one of these, remember that many proteins have quaternary structure. They have several protein subunits that come together. So once we get one substrate bound, it stabilizes the active conformation in all of the subunits, and we call that cooperativity. That's a mouthful. All right, and this is an example. It's a good example to take a look at. Compartmentalization is boring, but it's the kind of thing that AP might test you on. This is the idea of back from our unit 2 that we have organelles that compartmentalize different reactions. So for example, the lysosome is the stomach of the cell. It has digestive enzymes in it. We want those enzymes compartmentalized in one area so that they're only digesting the things that we want them to digest and not the whole inside of the cell. You might remember actually that the lysosome can break open, and that can lead to apoptosis and the death of the cell, and that can be a good thing. All right, you definitely need to be familiar with these graphs. They're going to be so important. So if you take a look at this particular graph, this is environmental impacts on enzymes. So the liver lab, we also saw in the spinach leaf disc lab, we saw the impact of temperature. But if you take a look at this black line right here, the peak of the black line, this is for a human enzyme, is at 37 degrees, and that's our normal body temperature. When you increase the temperature beyond, in Fahrenheit, it'd be like beyond 108 degrees, so very high fever, the proteins denature and they totally don't work. And remember, with hypothermia, where the temperatures are colder, the molecules slow down, but they don't denature. And so the proteins, if you warm the person up again, you can often return them to normal functioning. Whereas somebody who's had an extremely high fever, if their proteins have denatured, those don't always renature. And so sometimes that can be permanent brain damage, permanent blindness, deafness, all kinds of things. All right, and then pH is something that has an impact. So remember, pH is the hydrogen ion concentration. And so a protein has hydrogen bonds in it. And so if you change the pH from what it's supposed to be, you're going to change the conformation of any enzymes that are there. So the enzymes that are in the stomach, they're designed to be near lots of hydrogens, to have a high hydrogen ion concentration, aka a low pH, and that's the correct conformation. But if you start popping lots of antacids and changing the pH, then you can actually affect the digestive enzymes of the stomach. And same thing for trypsin, is an enzyme that's in the intestines, and it likes a slightly more basic environment. Last thing, same idea. So these are the same types of graphs here. The one that we didn't talk about, I'll make it a little bit bigger, is the idea, another environmental impact is how many substrates there are. So as the substrates increase, so does enzyme activity. They have more and more that they can work on. There is a point, however, called the saturation point, where you have so many substrates and not enough enzymes that we can't go any faster. The enzymes can only go so fast. And so then it levels off. It doesn't increase anymore. All right, I think that's it for enzyme. Oh no, there was one more thing, feedback. That's right. We're going to learn more about feedback later, but let me just walk you through it really quickly. Here's a substrate. It interacts with an enzyme to cause a whole series of reactions, and we end up producing a product called isoleucine. Isoleucine attaches onto an allosteric site of the same enzyme and shuts the reaction off and says, okay, we have enough isoleucine, we don't need to produce any more. until the cell uses up the isoleucine, and then it falls off the active site. The enzyme returns to its original conformation, and we make some more isoleucine. So brilliant, right? How your body works. Okay, that's it for enzymes. Let's talk about cell respiration. It's going to get heavier as we go. All right, so cell respiration, let me move through. I'm going to jump through what I think are the most important things. La, la, la. All right, this is a super important one. This is that whole idea about controlled reactions. Imagine this is the one where we use the example of a gas tank in your car and us dropping a match in the gas tank. So here's your gas tank, this reaction right here. We drop a match in, it explodes, and your car goes about 10, 20 feet, and we waste a ton of heat and light and sound energy. We just wasted lots of energy. Instead, here's your gas tank. Here are your spark plugs. We just cause tiny little explosions in your engine, really, really little explosions that are controlled, that don't blow up your vehicle and allow your vehicle to travel hundreds of miles. That's what your body does during photosynthesis, and you don't do photosynthesis, but cell respiration. We're controlling these reactions, only allowing a little bit of energy released at a time to control it as much as possible. If we released all of the energy at one time, our body temperature would increase beyond 108 degrees Fahrenheit, just as an example. Our proteins wouldn't be able to handle it, and that would all be a disaster. All right, so this is why we control the reactions the way we do. Okay, redox. We need to be really clear on the redox reactions. So remember, whoops, I'm going to go back to this one. Remember that when an atom or a molecule gives electrons, it is oxidizing. And when it receives, it has been oxidized. When it receives electrons, it has been reduced. You should write that down. If you're getting it all mixed up with oxidation and reduction, have it on a Post-it note, have it close to you. All right, next, and then the electron shuttles. So there are three primary electron shuttles that we studied in this unit. NAD plus, converting or becoming NADH plus H plus. FAD, and it becomes FADH2, and then NADP plus. That's the one that's in photosynthesis. These are the molecules that are going to catch electrons and deliver them somewhere else. And then they're going to come back. They get recycled. So they're going to catch electrons and deliver them, catch electrons and deliver them. And they keep recycling over and over again. All right. And then those are the two from cell respiration. So then remember, this whole diagram is supposed to represent the entire mitochondria. So the red is the mitochondria. Oh, I'm sorry. The red is the cell. Here's your cytoplasm. And then the yellow is the mitochondria, the outside of the mitochondria. The blue box is the inside of the mitochondria. And remember, the juice inside the mitochondria is called the matrix. And the space in between the two membranes of the mitochondria is called the intermembrane space. So let's go ahead and review some things. Cell respiration is a catabolic process. Catabolism refers to breaking things apart. For some dumb reason, I always think of, when I'm trying to remember the difference between catabolic and anabolic, I think of, so catabolic breaks things down. So I imagine a cat breakdancing. Does that help? I don't know if that helps, but cats breakdance, so they don't. Anyway, so catabolic reactions are breaking things down. So cell respiration is a perfect example of a catabolic reaction, because you have glucose that is an extremely organized molecule, and we're breaking it down into 6 carbon dioxide molecules. It is releasing energy. Remember, the whole point of this is that the plant stored the sun's energy in this glucose molecule. Now we're going to break that glucose molecule apart, just like the spark plugs in your car. We're going to do it a little bit at a time, and we're going to release just a little bit of controlled energy to do important reactions in your body without wasting energy and without raising your temperature. All right. This should just be a familiar diagram. In fact, you should really be able to just draw this diagram by heart. The one part of this diagram, I mean, there's a bunch of things that are details that are missing, but one thing that maybe I'd really like to see in this diagram is where carbon dioxide is being released. Glycolysis breaks glucose into two pyruvates, and then right here, carbon dioxide gets released, but there are two of them. There are two pyruvates, so two carbon dioxides get released. And then the citric acid cycle, so that was two of our six carbons from glucose. And then the citric acid cycle releases two carbons, but it goes around twice because of the two pyruvates. So that's our six carbons right there. So we've released all six carbons in that. And then the electrons are carried. So we produce a little bit of ATP there, a little bit of ATP in glycolysis. The majority of the ATP, something like 32 to 34 ATP molecules, are made in the electron transport chain during oxidative phosphorylation. All right, so let's go through the whole process, and hopefully this is very familiar. Review your small group assignments, because that's where I think the best diagrams are. Some of these diagrams get a little technical, and I don't think that they're necessary. So remember, the first thing in the cytoplasm, glycolysis takes a glucose molecule and it breaks it into two 3-carbon molecules called pyruvate. It takes 2 ATP, it produces 4 ATP, so overall you have a net of 2 ATP. You don't need to know an ounce of this. I'm going to bounce past it. Take a look. Actually, I'm going to go a little bit further. We'll come back to that. Substrate level phosphorylation. I don't think they're going to ask you specifically what substrate level phosphorylation is. However, they might use that phrase on a test. So remember, this is in contrast to the last stage of cell respiration, which is called oxidative phosphorylation. So substrate level phosphorylation is when you have an enzyme and you're taking a phosphate from an organic molecule and attaching it onto ADP to make ATP. So substrate level phosphorylation happens in glycolysis and it happens in the Krebs cycle. The oxidative phosphorylation that happens during the electron transport at the very end, that's happening from an inorganic, not an organic, from an inorganic phosphate. And so that one's got a different name. So that's oxidative phosphorylation. I don't know if I explained that very clearly, but I did my best. All right. Oh, and then glycolysis, we're making 2 pyruvate. We made 2 NADH, so we captured a couple of electrons. Whenever you capture a couple of electrons, which have a negative charge, usually a couple of hydrogen ions clamp on in order to balance the charge. And then our net is 2 ATP because we used 2, we produced 4, so our net ATP in glycolysis is 2. All right, so next thing that's going to happen, if there's no oxygen present, then we're going to go into fermentation. Remember just roughly that at the very end of the electron transport chain, oxygen is catching the electrons. If oxygen isn't there to catch the electrons, it's like a plug in a pipe and the whole system backs up. The NADH and the FADH2s, they can't drop off their electrons. They can't recycle and catch electrons. So the whole system stops. The Krebs cycle stops. And the only thing that can really happen is glycolysis and fermentation. If oxygen is available, then the whole process can move forward. So let's remind you really quickly what fermentation is. I'm just going to jump ahead. The 2 types of fermentation that you need to be most familiar with, there's a bazillion other types. Ethanol fermentation, which is what happens in yeast and where we get alcohol from, and lactic acid fermentation, which is what happens inside your cells when your cells are not getting enough oxygen. Sometimes that might be because you're exercising and you're just not getting enough oxygen. Sometimes it might be for some disease process. All right. Yeah. And so they still release carbon dioxide. So you're releasing some of the carbon dioxide here. The interesting thing is that ethanol and lactate can be reused by the body once oxygen is available. What you definitely need to know about both processes is that they make 2 ATP. Nowhere near enough ATP for a human being to survive on. We need so much more energy than that. A yeast cell can survive on just ATP. So fermentation works just 2 ATP. So that works for a yeast cell, but not for us. All right, so let's assume, we're going to be right here. Let's assume that we have oxygen and we're going to start moving. Glycolysis is going to break up those pyruvates and move it into the mitochondria itself. So here's the pyruvate. It goes into the mitochondria. I feel like your small group activities have the best diagrams, but this is a pretty good one right here. The pyruvate from the glycolysis is brought in. We lose 1 carbon dioxide. However, there were 2 pyruvates, so we've lost 2 carbon dioxides. And then this diagram here, I also like. As it enters the citric acid cycle, we lose 2 more carbon dioxides. But again, there's 2 of those acetyl-CoAs entering, so it turns twice. So we actually lose 4 carbon dioxides. So this is where each carbon is pulled off of the glucose and they're eliminated as carbon dioxide. That's our waste product. In the process, very, very importantly, let's see if I can get this to move up a little bit. We make 3 NADHs and we make 1. I got to say it right because the numbers are sometimes confusing. 1 FADH2. However, it turns twice. So really, we get 6 NADHs out of the deal and 2 FADH2s. Those are our electron shuttles. They're going to carry the electrons that they just got over to the electron transport chain and drop them off. We are doing these tiny little shock absorber, that's the wrong thing, spark plug reactions to control the release of energy. I'm going to move forward. Now this citric acid cycle diagram, it's just too complex. Really look at your small group assignment. I feel like it's just a much better diagram for what you need. Actually, there was one other thing I wanted to just remind you. Give me a second. Oh, it doesn't have it here. I thought it did. Okay, well, we're good enough. I think we've talked enough about the citric acid cycle. Sorry if you're getting busy watching this. So let's go and focus in on, this is actually my favorite diagram on this. And so I just want to make sure that you are comfortable with this diagram. So during the Krebs cycle, we made those 6 NADHs and 2 FADH2s. There are electron shuttles. They're now going to drop off these high energy electrons. Now they don't necessarily look high energy here. This diagram, which is the same diagram, but it's drawn differently. They're drawing the same thing. Notice the NADHs dropping off their electrons, and the artist drew it in a way that shows you that the electron is gradually losing energy as it moves down. And as it loses energy, that energy that's being released is being used. Come on, it's hard to get these prezzies to go where I want them to go. There we go. That energy that's being released is being used to pump hydrogens into the intermembrane space. The pH drops while the hydrogen ions increase in there. And then the hydrogen ions then start moving through the enzyme. The one enzyme you absolutely need to know the name of is ATP synthase. Now, this is a super simplified drawing of ATP synthase. It's a turbine that as the hydrogen ions move through, it attaches the inorganic phosphate to the ADP and we make ATP. This is oxidative phosphorylation, not substrate level phosphorylation. Other important things in this diagram. Notice that oxygen was at the end and it caught the electrons, and then because it's got 2 extra electrons, right away 2 hydrogens join it and we've made water. If oxygen isn't there, there's nobody to catch the electrons. The whole process stops and we have to go into fermentation. And then the last thing I guess I haven't mentioned is just The name chemiosmosis osmosis is the moving of the hydrogen ions through ATP synthase, and it's a chemical instead of water. That's what it's referring to. All right. This is an image of ATP synthase. Whoops. Just showing you sort of the turbines, but I don't think you need to know anything more than that. Don't care about dead white guys. I mean, I care about them deeply, but in this process, we make about 32 to 34 ATP. Other important stuff. So the final accounting, remember that in your student activity assignment, it said you can make up to 38 ATP. But the reality is that we never make that much, and that's because cells just are inefficient. They're much more efficient than any human-made machine, way more efficient. But there's always going to be some release of heat and some wasted energy. So we don't get that full 38 ATP. Roughly, we get 3 ATP for every NADH, and we get 2 ATP for every FADH2 that gets made. All right, and then moving on, a couple other things. This again was mentioned in your small group assignment. We focus on glucose when you're first learning about cell respiration, but you need to understand that you don't just eat glucose, you eat proteins, carbs, and fats. And so those all can get incorporated somewhere in this process. Here's glycolysis, here's the citric acid cycle, here's oxidative phosphorylation. At some point along the pathway, any of those food types can be used. We just focus on the carbohydrates initially just while you're learning. And then, yeah, that's enough. I don't think we need to know anything else. I think we're done. Let me just double check. Yep, we are all done with this one. Last one, everybody. We need to talk photosynthesis. Still the big one, I'm sorry. All right, so remember on the diagram of photosynthesis that what you're looking at here, give me just a second. This orange is the chloroplast itself. The blue is oh my gosh, I'm trying to remember how this works. The blue is the green is the thylakoid, the blue is the chloroplast, and the orange is the cell itself. That's what it was. I was trying to remember how they drew this diagram here. All right, let's jump in. It is not moving. It makes me sad when it doesn't move. There we go. Now it's working like crazy. Let me jump way back because it went super fast. All right, let's try it again. Moving in, important things that I want you to know. First of all, I want you to know the anatomy of a plant. So you really need to know like these terms in this. Oh my gosh, really? I'm sorry, you guys. I don't know what just happened. You need to know these terms. So you've got your chloroplast. Inside the chloroplast, the little green discs, those are the thylakoids, and that's where the light-dependent reactions are occurring. And then a stack of them is called a granum or grana. And the stroma is the liquid. What was the liquid in the mitochondria called? Matrix mitochondria. Stroma is in the chloroplast. Oh, and one other thing that I wanted to make sure you remembered is that the stomata are the little openings that you find on the bottom of the leaf. Remember that the leaf has an upper epidermis and a lower epidermis. The upper epidermis tends to get the sunlight on it. So the stomata are on the bottom where it's more shaded and less water evaporates outside or out of those pores. And those are the pores that let carbon dioxide in and oxygen out. All right, information about light energy. Well, I'm not going to go over a whole lot. Chlorophyll is a pigment. Pigments are things that react to light, and we'll talk about that in just a second. So chlorophyll is the primary pigment that we're concerned with, but there are other pigments, and you see those other pigments in the fall when chlorophyll begins to leave. You start to see some of the carotenoids and the xanthophylls, which have different absorption spectra. We'll review that in just a second. And that makes the leaves look a different color. All right, so sunlight contains almost all wavelengths of light. So why do plants look green? And I'm guessing that most of you remember this, but I think this is really important. Like, I can almost guarantee you that these images are going to be on a test, if not my test, on some other test. So these are called absorption spectra. The higher it is, the more it absorbs the particular wavelength of light. So for example, this chlorophyll B really likes to absorb light at what, about 450 nanometers, but then almost nothing in the green area of the spectrum. And then again, it starts to absorb in the orange, and the other one absorbing in the red range. So because it's not absorbing green light, you see the green light. You can see the same process coming over here. When green light passes through chlorophyll, it passes all the way through and your eyeballs can detect it. When blue light gets passed into the chlorophyll, it gets absorbed and you see nothing. Nothing passes through it. The other thing that I wanted to show you on this one, because I think it's such a good experiment and definitely AP kind of questions can come from this. So this was Engelmann's experiment where he grew bacteria on algae that was photosynthetic. So this algae can do photosynthesis, and the bacteria are aerobic. Now that's super important. That means that they like oxygen. So they're going to grow in areas where there's lots of oxygen, and they're not going to grow in areas where there's not much oxygen. So what Engelmann realized was that the bacteria were growing in large amounts in the purple and blue areas of the spectrum and in the red and orange areas of the spectrum, but very little bacteria was growing in the green areas of the electromagnetic spectrum. And he realized that that was an indicator that more photosynthesis, producing oxygen, these bacteria need oxygen, was happening in these color areas, and very little photosynthesis was happening here. That's because the green light is being reflected away and not being absorbed and not doing any, not allowing the plant to do photosynthesis. So I think that that's a cool little experiment and definitely AP worthy. All right, so let's review. Anabolic versus catabolic. Remember cats break dance? I don't know. Catabolic is breaking things down. Anabolic reactions is putting things together. We're building molecules. The whole idea is that you're taking in, the plants are taking in the sun's energy to store it by building glucose and other molecules. So it requires energy, and that makes it endergonic. All right. We already went over that. This is a great summary of everything that's happening. Light coming in, water is needed. These are the light reactions. And then ATP is made and NADPH is made. It's carrying some electrons over to the Calvin cycle that is doing the carbon fixation. It takes a little bit of ATP to fix that carbon, but then we're storing it in these big molecules of glucose and cellulose and all kinds of other molecules. All right, so that's kind of the summary of it. Let's go into a little bit more detail. Remember that when light hits a chlorophyll molecule, a pigment molecule, it gets absorbed. That energy gets absorbed, and it causes an electron to get excited. It's absorbed the energy, and it jumps from one level to the next. Usually, the electron comes back down to its original level, and when it does, it releases a photon of light or some heat or a combination. That happens in this diagram here. You see electrons constantly rising and returning and rising and returning, and photons being released and energy being released until you get to the special reaction center that has the chlorophyll A molecules. The chlorophyll A molecules eject the electron, and it's actually gone. It's captured by the primary electron acceptor. So they are now missing an electron or two electrons, because there's two of them. So who donates the electrons? Water. Okay, so let's go to my favorite. And this is, don't forget, just reminding you really quickly, this one, let's see if I can see it here. So P680 is the one for photosystem 2. Remember, photosystem 2 comes first, and photosystem 1 comes second, and that one has P700, and that refers to the wavelength of light that they like to absorb the most. This is my next favorite diagram. I think it's just a really clear diagram. Photosystem 2, light photon. It's passed from one pigment molecule to the next until it gets ejected in the reaction center. Water is broken apart. It donates its electrons. It's reducing it. It's oxidizing, so it's donating the electrons to the chlorophyll. So now whatever electrons were missing have just been replaced. The electrons that got ejected then are handed down an electron transport chain. As they move from high energy to lower energy to lower energy, just like in the mitochondria, the cell is using that energy, very controlled little explosions, to pump hydrogens into the thylakoid space. The pH level goes down because the hydrogen ions are up. Those hydrogen ions then diffuse back through, oh, look at that, ATP synthase again. And this turbine ATP synthase is making ATP. Why are we making ATP, KJ? I thought we were making glucose. We are. We have to make a little bit of ATP in order to do the carbon fixation. In order to store a ton of energy, we have to use a little bit of energy. In addition, there's photosystem 1. So as these electrons get passed on, they get captured by photosystem 1, which has ejected its electrons. Light is powering it again, more photons, more energy being added to the system. The electrons get ejected and eventually captured by NADP+, which turns into NADPH. If you have any trouble remembering NADH and NADPH, the P1 is in photosynthesis. So the one that has phosphorus in it is in photosynthesis. Okay, both the NADPH and the ATP are then going to the Calvin cycle. I'm going to move on. This one is a little bit, I don't want to talk a whole lot about it, but this is what is supposed to be happening. This is called non-cyclic electron flow. So we're getting new electrons from water and we're dumping them off over on this end. It is possible, if there's no water available, to have something called cyclic electron flow. In that case, we make a little ATP, but we're not storing it up and storing up the sun's energy in glucose or whatever. And of course, that's not happening in us. It's happening in plants. All right, great analogy, definitely worth taking a look at. Right here, so after we're done with this process, we've made a little bit of ATP, a little bit of NADPH, and we were releasing oxygen here. Remember, we ripped the electrons off of water, and so now oxygen is being released. So this is where oxygen is being produced in the whole photosynthesis process. Okay, so now, let me back here. Now we're going to start bringing in carbon dioxide. This is called carbon fixation. And this is where we're going to take a low energy molecule, carbon dioxide, and we're going to start putting them together to build a high energy molecule like glucose. I think that this diagram is unnecessarily complicated, and your diagram on your student activity sheet is better than this. There are 3 phases. The one you absolutely need to know is the phrase carbon fixation, the idea of removing carbon from the atmosphere and attaching it to others. So that's what's happening, and it's being captured by an enzyme called rubisco, super important enzyme, and we're going to talk about it in just a minute. It also likes oxygen, which can screw things up. All right, and then just your student activity sheet did an awesome job of kind of giving you an accounting of all the different carbon atoms. Basic idea, if I were going to say it super fast, for every 3 carbon dioxides that come in, then a molecule that has 3 carbons on it can be released. Now, that doesn't make sugar, KJ, because glucose has 6 carbons, C6H12O6. Well, if you put 2 of the 3 carbon molecules together, then you can actually build glucose with its 6 carbons. So you put them together. All right, moving on. Blah, 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 blah. Let's go to important things. Okay, this is about rubisco right here. This is the rubisco enzyme, and rubisco is supposed to be attracted to carbon dioxide, and it does an awesome job. However, it's really attracted to oxygen also. And so if levels of carbon dioxide are a little bit low, it will start binding to oxygen. That's called photorespiration, and that interferes and makes photosynthesis less efficient. So plants have evolved adaptations for that. Ideally, the plant leaves its stomata, the stomates open, and so we get lots of carbon dioxide in and lots of oxygen out, and so Rubisco works perfectly. But that's difficult in a hot, dry climate because not only does oxygen come out, but water comes out also. And so the plant starts to desiccate, it starts to dry up. So the plant has to close its stomata. Well, that causes an increase of oxygen on the inside of the plant, and then Rubisco starts binding with the oxygen instead of the carbon dioxide. That's a problem. So there's two adaptations for that. You do not need to know a whole lot about it. The C3 adaptation, sorry, the C3 is the normal. I'm like, what am I looking at? The diagram wasn't there. C3 is what we normally have. What the adaptations are for dry weather, hot, dry weather, there's the C4 leaves. All you need to know is that they are spatially separated. They're in two, the processes are happening in two different cells. Just trust me on this. So the light dependent reaction are happening in one cell. And then the Calvin cycle is happening in a different cell. And by separating them, they're able to keep the carbon dioxide levels high and the oxygen levels low. So that works for some plants. The other adaptation are called the CAM plants. In CAM plants, I think this one's genius. Instead of having their stomata open during the day, which allows water loss, they close their stomata and they open them at night when it's cooler and not as much evaporation is happening. So during the night, they open their stomata, they take in tons of carbon dioxide and they store it. And then they close their stomata during daylight, but they have all this stored carbon dioxide. And so this photosynthesis can still happen during the daylight. And we don't have to worry about photorespiration because we've got so much carbon dioxide that was stored during the night. All right, I think that's it, cuties. Yeah, we're all done. Oh my gosh, I wonder how long that was. I hope you have a super good rest of your day. Let me know if you have questions.